The next labs that we are going to complete are the diffusion and osmosis labs from your lab manual. The information that corresponds with the diffusion and osmosis labs will be from Chapter 5. The goal of this presentation is to provide you with information that you will need in order to complete and understand the diffusion and osmosis labs. Make sure that you copy all of the notes down that I present and write down, and you bring your handwritten notes to class. If you need to pause and write stuff down, please make sure that you do so in order to bring all of the information to class when we conduct the labs. Chapter 5 includes information that is about the cell's plasma membrane or the cell's membrane. Information that you will want to understand from Chapter 5 will be the anatomy and physiology of the plasma membrane of the cell. We will first start by describing the composition or the anatomy of the plasma membrane. The first thing to understand about the plasma membrane is going to be its composition or its anatomy. The plasma membrane is primarily made of substances called phospholipids. But there are other substances that you can find in the plasma membrane as well. The other substances that are embedded within the plasma membrane are proteins and cholesterols. The plasma membrane of the cell is similar to a very thin layer of oil. Another characteristic that you want to know about the plasma membrane is the plasma membrane is what we call selectively or semi-permeable. This just means that the plasma membrane is very selective about which substances that it will allow to cross the membrane and enter or exit the cell. The next task that we are going to do is we are going to draw a diagram of the cell and discuss some of the functions of the plasma membrane. So I'm going to go ahead and draw a cell at the center of the rest of the page, and then I'm going to write some notes along the edges of the cell. There's my cell. Please recall from chapter 4 that the inside of the cell is an ooey gooey wet environment, and that most cells are constantly submerged and exposed to fluids surrounding them. So let's go ahead and add some terminology to the inside of our cell. This is the intracellular area, or what we call the intracellular fluid inside of the cell. That is abbreviated ICF. Just outside of the cell is called the extracellular fluid. Which is abbreviated ECF. Please keep that in mind throughout the notes. The intracellular fluid is a water-based fluid with various substances that are called solutes. The intracellular fluid is just a mixed fluid that is water-based, containing many different types of substances like salts, sugars, amino acids. Any substance that is mixed in the intracellular fluid is going to be called a solute. So we have various types of solutes, including salts, sugars, amino acids, etc. Let's talk about some movement mechanisms across the cell's membrane. Examples of movement mechanisms across the membrane. The first movement mechanism across the membrane is pretty simple. Substances may directly cross the membrane to enter or exit the cell. The types of substances that typically can cross directly through the membrane are going to be small substances, hydrophobic or nonpolar substances. So small, hydrophobic, or nonpolar substances may be able to easily pass across the membrane to either enter the cell or exit the cell. I will provide some examples of small hydrophobic nonpolar substances that are able to easily cross the membrane to enter and exit the cell later through the presentation. Let's talk about another movement mechanism across the membrane. So another example of how substances enter or exit the cell by crossing through the membrane. Let's talk about water, since water is essential to life. Water may enter or exit the cell when they cross through a type of protein called an aquaporin. An aquaporin is a protein that is going to be embedded in the membrane. And water will be able to enter and exit the cell through the aquaporin. There's water entering or exiting the cell. Let's talk about the next mechanism. Some substances have to move through a protein that is embedded in the membrane in order to enter or exit the cell. Examples of these substances are larger substances, for example, sugar molecules. I will give more examples of these types of substances later on in the presentation. So some substances move through a membrane protein. Let's just draw that membrane protein right now. Usually a fairly large substance. That's the membrane protein. And the reason that the substances move through a membrane protein is because they are not compatible with the chemistry of the plasma membrane. Recall that the plasma membrane, this lining right here, the plasma membrane, is a thin layer of oil, and not all substances are compatible with oil. Substances that are not compatible with oil have to move through what's called a membrane protein. Examples of substances that have to move through the membrane protein to enter or exit the cell are going to be large substances or hydrophilic or polar substances. So notice that these substances are completely the opposite of the substances up here, small, hydrophobic, and nonpolar substances, which can very easily cross through the oily plasma membrane because these substances tend to be compatible with the oily layer. We have two more things to talk about. Let's talk about vesicle transports. Another mechanism that the cell can use to either allow substances to move into or out of the cell is a bulk transport mechanism called vesicle transport. A vesicle is like a little round pouch in the cell. This is my vesicle. And the vesicle contains substances. I'll just put some dots inside of the vesicles indicating that the vesicle contains substances. This could be various different types of substances. And I will give an example later on in the presentation. What happens is that the vesicle will fuse with the membrane, and then basically that fusion is going to allow the substances to exit out of the cell. Then the substances are released to the ECF. Please recall that the ECF is called the extracellular fluid. The last mechanism that I would like to talk about is a little bit more complicated, and it includes something called a receptor. Let's please go ahead and mention this and draw a picture next to it. Oftentimes, a plasma membrane will have a protein that is basically on the surface of the plasma membrane. This protein may be slightly embedded in the plasma membrane, but right now, let's call this protein that is on the surface a receptor protein. Let's talk about one function of a receptor protein. Receptor proteins may have many functions, but its primary function is to bind chemicals to the receptor portion of the receptor protein. So the receptor protein binds chemical messengers, or basically chemicals that may be found in the extracellular fluid. When that chemical messenger binds to the receptor protein, then a cell activity is triggered. Chemical messengers are often called hormones. Binding causes specific cell activity. Let's draw another receptor protein, the same as the first one. The protein is found at the surface of the cell, and floating around in the extracellular fluid is a chemical messenger. What happens is that chemical messenger will move into the binding site, and once the binding occurs, it's going to trigger the cell to do some sort of activity. And this is why hormones are called chemical messengers, because when they bind to the receptor protein, that ultimately delivers a message to the cell to command the cell to perform a specific activity. For example, if this was insulin, then when insulin binds to the receptor protein, it is going to command that cell to absorb extra sugar out of the blood. The major function of insulin in the body is to communicate with cells and tell cells to absorb excess sugar out of the blood in order to bring the sugar levels in the blood back down to normal levels. The next major topic that we want to discuss is diffusion. We need to discuss diffusion because we are talking about how substances are moving into or out of the cell. Diffusion is a major concept that describes the movement of molecules and thus the movement of substances into and out of the cells. The best example to give for diffusion is a perfume bottle. When you have your perfume bottle, the perfume molecules are concentrated inside of the bottle. And as soon as you spray the perfume bottle, some of the perfume molecules are released and they spread apart in the air. Spreading apart of molecules is called diffusion. Let's go ahead and list a more scientific definition of diffusion in order to make sure you are comfortable with understanding diffusion. Diffusion is the net movement of molecules from high concentration to low concentration. The net movement of molecules from high concentration to low concentration. The brackets indicate concentration. The net movement of molecules from high concentration, so these brackets mean concentration, to low concentration. That's the same as saying the perfume molecules spray. So they spread apart. Spreading apart is a less complicated way to describe diffusion. Molecules tend to spread apart, or you can say molecules tend to diffuse. There are certain factors in the environment that could affect the rate at how fast that molecules diffuse. Let's talk about the first factor in the environment, temperature. Imagine that you have a pot of boiling water. Do you think the molecules are moving fast or slow as the water heats up? The water molecules are moving faster as the temperature increases. So when there is a high temperature in the environment, molecules tend to diffuse at a faster rate. What about if we have a decreased temperature? Well, that will end up decreasing the rate of diffusion. The next factor that affects the rate of diffusion is the size and the weight of the molecule. A smaller lighter molecule is going to move faster, therefore it will diffuse quicker. If you had a heavier, larger molecule, it is going to end up diffusing slower. The third factor is the type of media through which the molecule is diffusing. The best way to describe this is imagine that you have a cup of water on your kitchen countertop, and you also have a cup of Jello. Except imagine that it's not the fun Jello. Imagine that it is clear Jello, like we have in the lab. So in one cup with the water, that represents a liquid media type. And in the other cup with the Jello, that represents a solid media type, right? Because water is liquid, and Jello is solid. Imagine that you take some food dye, you know how they come in dropper bottles, and you drop food dye, one drop of food dye on both the liquid and the Jello at the same time. Do you think the food dye molecules will diffuse faster through the water, which is the liquid, or Jello, which represents the solid? The food dye is going to diffuse a lot faster through the liquid compared to the solid media. So the three types of media, because we're not in chemistry, we can keep it simple. The three types of media are going to be solid, liquid, and gas. So remember, our example of liquid was the water in the cup, and our example of solid was the Jello. When you think of the gas media, just think of air. The rate of diffusion is going to be the highest in the gas medium. Our last factor that's going to affect the rate of diffusion is called steepness of the concentration gradient. Let's discuss the concentration gradient on the next page, but go ahead and make a note here. A steeper gradient, or a higher gradient, is going to equal a higher rate of diffusion. And we're going to give an example of that on the next page. On the next page, we're discussing what is a concentration gradient. A concentration gradient is the difference in the amount of a substance in two different areas. So two different areas that we discussed at the beginning of this presentation would be the ICF and the ECF. If you do not know what the ICF and the ECF are, please go back and look at the first page of this presentation. Two examples would be the ICF versus or compared to the ECF. For the next question, let's draw a picture and make some notes around it, which will help us understand what the concentration gradient is and how a steeper concentration gradient increases the rate of diffusion of a molecule. I am now going to attempt to draw a beaker. There's my beaker. I'm going to add some fluid into my beaker. I'm going to call this fluid the ECF. I will drop a cell into the fluid. So now my cell is completely submerged in the ECF. So let's say that the cell is composed of 50% water. So there's 50% water inside of the cell. This is not typical for cells. I'm using this as an example. And the ECF was composed of 100% water. So we have two different areas. We have the ICF, which is inside of the cell, and we have the ECF. The ICF contains 50% water, whereas the ECF contains 100% water. So just like the concentration gradient states, we have a difference in the amount of substance in two different areas. We have a difference in the amount of water in two different areas. According to diffusion, substances will move from an area of higher concentration to lower concentration. Therefore, water, which is higher out here, there's 100% out here, is going to diffuse into the cell. Water diffuses in. Why does water diffuse in? Because there is a concentration gradient. Let's determine the value of the concentration gradient. You're basically going to take the bigger number and you're going to find the difference between the bigger number and the smaller number. So 100% minus 50% equals 50%. There is a 50% concentration gradient between the two different areas, ICF and ECF. Recall that when you have a steeper concentration gradient, that is going to increase the rate of diffusion. So an increased concentration gradient will result in an increased rate of diffusion. So water will move from an area of higher, which the area that contains the higher amount of water is the ECF, to an area of lower concentration gradient, which is inside of the cell, the ICF. Notice on the next problem that it says, we will do this part in class because we're going to look at a specific portion of the diffusion lab, which is part 3, And you're going to go ahead and apply what you know about diffusion to this lab. Make sure that you bring your handwritten notes to class so that we can complete letter C together. I will make some notes on the board for you. Since water is so important to living things, we have a term that describes the diffusion of water specifically. That term is called osmosis. So osmosis is going to be the diffusion of water specifically across what we call a semi-permeable or selectively permeable membrane. In our previous example, where we dropped the cell in the beaker of fluid, water was allowed to diffuse across the membrane. Recall that not all substances are allowed to move across the membrane. This is what makes the plasma membrane selectively or semi-permeable membrane. Let's go ahead and note that not all substances can cross easily the membrane. The term for the substances that typically have a more challenging time crossing the membrane are called solutes. When you think of a cell in your body, do you think the cell is sitting in a dry environment or a wet, gooey environment? Most cells are constantly exposed to and submerged in gooey fluid environment. And those extracellular fluids that are surrounding the cells may affect the cells. The next topic that we're going to talk about is how the extracellular fluids affect cells, or how different extracellular fluids affect cells. This definition is basically saying that the extracellular fluids that are surrounding the cells can impact cells in various ways depending on the solution. There are three different types of solutions, and they each have different impacts on the cell. The first type of solution is called isotonic. That term tonic means solute, iso means same. An isotonic solution or an isotonic fluid is a type of extracellular fluid that has the same concentration of solute in the ECF and the ICF. Remember, the brackets mean concentration. The second type of solution is going to be called hypertonic. And a hypertonic solution will have a higher concentration of solute in the ECF compared to the ICF. And the last one is called hypotonic, and it's going to be the opposite of hypertonic. A lower concentration of solute in the ECF compared to the ICF. In order to more understand this, I am going to include a picture. And the picture is going to be for the first one called the isotonic solution. Term iso means same. The term tonic means solute. So we are going to draw a beaker with a cell dropped inside, just like we did in our previous example. Recall that the inside of the cell is called the ICF, and the fluid surrounding the cell is called the ECF. Well, let's say the ECF is isotonic compared to the ICF. The cell in this example is composed of 97% water. That means that the other 3% are those various substances that we call solute particles. We have to account for 100% of the ICF. So therefore, we have to say, okay, if there's 97% water, there must be 3% solute. Remember that the term isotonic means same concentration of solute in the ECF compared to the ICF. So if it's the same concentration of solute, that means the ECF should also have 3% solute and 97% water. Just giving hypothetical examples about cell compositions. So 97% water and 3% solute is not realistic for cells, but we are trying to understand some basic concepts. Another thing that we want to note is that the plasma membrane, which is the lining right here, is semi-permeable. This means that it will allow water to cross. So water can always cross the plasma membrane, but solutes may not be able to pass the plasma membrane. So solutes may not be able to cross the membrane. Is there a concentration gradient? Is there a concentration gradient? Well, how much water is inside and how much water is outside? 97% minus 97% equals 0. So there is no concentration gradient in this example. And if there is no concentration gradient, then there is no force to drive a net diffusion. So if there's no concentration gradient, then there is no force to drive diffusion. This means that water must be in what we call equilibrium. It is equal in both the ICF and ECF. This is actually called dynamic equilibrium because the water is floating in and out of the cell, but it is always balanced across the membrane. We have to move on to the next one, which is hypertonic. And that term hyper means more, and tonic means solute. So if you look back at your definition of hypertonic from the previous page, you will see that the definition of hypertonic is an extracellular fluid that has a higher amount of solute in the ECF compared to the ICF. Again, we're going to go ahead and draw the beaker. With our cell. The ICF is going to be the same in this cell as it was in the last cell. So let's go ahead and copy that down. Recall that there's 97% water and 3% solute in our last example. We're going to keep that the same in this example and the next example. Our extracellular fluid is what we call hypertonic relative to the cell. Hypertonic means there's more solute in this extracellular fluid compared to the amount of solute in the intracellular fluid. So, for example, 5% solute. That is more than what's inside of the cell. If there's 5% solute, then how much water is there? 95% water. Please do not forget that the plasma membrane is semi-permeable. It allows water to pass, but solute cannot cross the membrane. Again, the goal behind this is to understand some concepts. So solute cannot cross the membrane, but water can cross. And just like all molecules, water will want to move from an area of higher concentration to an area of lower concentration. Can you identify where there is a higher concentration of water? It looks like there is a higher concentration of water in the ECF. And then there is a lower concentration of water in the ICF. And water will always want to move from an area of higher to lower. So which way is water going to move? Water will move from where it's high, 97%, to where it's lower, 95%. So it's going to move from the 97% outside of the cell towards the 95%. So water will diffuse out. So in this case, when cells are exposed to hypertonic solutions, water moves out of the cell. This process is called osmosis. This will affect the cell's volume and the cell's shape. As more and more water diffuses out of the cell is going to have less volume, and it's basically the cell is going to shrink up. In plants, when a cell shrinks, so in plants, when a cell shrinks, that process is called plasmolysis. Plasmolysis or plasmolysis. This is similar to when the plant begins to wilt. In animals, and specifically, let's talk about a type of cell called an RBC, which is red blood cells. Shrinking cells are called crenation. And the problem with blood cells that are being crenated or that undergo this process called crenation is that once the shape changes and the cell begins shrinking, it's not able to bind oxygen or move effectively through the circulatory system. So this is definitely a very big problem if blood cells are exposed to hypertonic solutions. We're going to talk about our last one. We're almost there. Our last one, our last type of solution is called hypotonic. And that term hypo means less, and tonic means solute. So less solute in the extracellular fluid. Again, I'm going to draw a beaker and drop a cell inside. I will keep the composition of the cell the same, 97% water and 3% solute, the same as in the previous examples. So what if the fluid surrounding the cell, that fluid that's surrounding the cell, is hypotonic? Can you give me a number value for the amount of solute that might be in the ECF if the ECF is hypotonic compared to the cell? Anything less than 3 would be your answer. So let's say there's 2% solute in the ECF. How much water would there then be in the ECF? That would be 98% water. Has to equal to 100. Can you identify the area that has a higher concentration of water? It looks like there's 98% water in the ECF and only 97% water in the ICF. So we have a difference in the amount of substance in two different areas. Do we have a concentration gradient? Is there a concentration gradient? So in order to determine if there's a concentration gradient, you take the higher amount of water in one area and you subtract the lower amount of water. So 98% minus 97% is a 1% concentration gradient. And anytime there is a gradient, that means that there is a force to drive diffusion. So which way will the water diffuse? Please do not forget that the plasma membrane is selectively permeable, or what we call semi-permeable, and in this example will block solute. So even though there is a difference in the amount of solute, the solute will not be able to diffuse across the membrane. This could be due to the pores in the membrane being very small and simply not being able to allow large solute molecules to pass through. Water is a really small molecule and can typically pass through almost all membranes. So even though the plasma membrane is selectively permeable, water always has a free pass to cross. So it looks like, since water is going to move from an area of higher to lower, that water is going to move into the cell. So water diffuses in. And this is going to increase the cell's volume. The cell's volume increases. So the cell swells and could pop. The cell may possibly burst depending on what type of cell it is. Animal cells would burst. Animal cells could burst because they could intake so much water that eventually they will basically burst open. And this is because animal cells are not confined by a thick lining called a cell wall. Cell walls are very thick linings surrounding and protecting cells. When red blood cells of animals are exposed to hypotonic solutions, they are at risk to burst. The bursting of a red blood cell is called hemolysis. Plant cells are a little bit different because plant cells are surrounded by a thick non-living matrix called a cell wall. The cell wall will prevent the plant cell from popping. Instead, the plant cell will intake so much water that it will exhibit pressure and press up against the cell wall. This pressure is called turgor pressure. So plants actually like to be exposed to hypotonic solutions because when the plant cell exhibits turgor pressure and presses up against the cell wall, this gives the plant organism as a whole structure. For example, if a plant is growing taller and taller to be exposed to sunlight. Okay, let's move on to the next one. We are almost near the end. We have a couple of pages left. Let's discuss some major mechanisms of movement across the membrane. We talked about it a little bit on the first page of our notes in this presentation, but we need to continue and more specifically identify the names of these types of movement across the membrane. The first major mechanism for how substances move across the membrane is called passive transport. Substances may use passive transport to cross the plasma membrane in order to enter or exit the cells. Passive transport is going to be when a substance diffuses into or out of the cell. This means the substance is typically moving from an area of high concentration to an area of lower concentration. And when we have a situation in which substances are basically in an area of higher concentration compared to lower concentration, molecules want to tend to spread apart on their own. They use their own molecular energy in order to diffuse. So anytime a substance diffuses, we say that substance is using passive transport. A couple things to note about passive transport. Anytime you say a substance diffuses, that's going to be passive transport. Anytime you say a substance moves from an area of higher to lower concentration, that's going to be passive transport. In addition to those characteristics, we want to make sure that you understand that cells do not have to use any of their precious energy molecules to support this movement. So this process does not require precious cellular energy. There is a molecule called ATP, which is the cell's main energy molecule. This molecule is not needed in order for passive transport to occur. Let's give some examples of transport, and we talked about a few of them already. The first one is called simple diffusion. During simple diffusion, substances diffuse directly across the lipid bilayer of the membrane. These substances are usually small, hydrophobic, or nonpolar substances. Small, hydrophobic, and or nonpolar substances. Let's give some examples. Carbon dioxide and oxygen gas. Take a deep breath in. Oxygen is going into your body, and oxygen is then diffusing into your blood and then going to be diffusing into your cells. Oxygen is actually a nonpolar type of molecule, and it is also a very small molecule. The oxygen molecules can diffuse directly across the membrane, and the diffusion process is really quick. This is very good for us because, of course, we want to continue to have oxygen being diffusing into our cells regularly. On the other hand, carbon dioxide is easily able to diffuse out of our cells and into the blood and then out of our lungs as the blood moves through the lungs. Carbon dioxide is a waste product for us, and we do not want it to accumulate in our bodies. So therefore, since carbon dioxide can easily diffuse out of us, this makes this mechanism called simple diffusion very, very important. And I also want to remind you that getting oxygen in and carbon dioxide out of our bodies does not require the cell to use any of its precious energy molecules. Oxygen and carbon dioxide basically move based on their own kinetic or their own energy because they are using diffusion. And diffusion by itself is a force which drives diffusion. A concentration gradient by itself is a force which drives diffusion. All right, the next mechanism to discuss is called facilitated diffusion. This is going to be when substances diffuse across a membrane through a membrane protein. These types of molecules tend to be really large molecules. They could also be smaller molecules too, but they're basically the type of molecules that are hydrophilic, which is the opposite up here, and polar type of molecules. Ultimately, these molecules are either large or they are not compatible with the phospholipid oily layer of the membrane. Recall that not all substances are compatible with oil. Think about at home when you have a cup of water and you add some oil and you mix it up, the oil separates from the water. Not all substances are compatible with oil. Some substances do not like oil, and those substances are called hydrophilic or polar substances. So the type of substances that utilize this mechanism called facilitated diffusion are substances that are large, hydrophilic, and or a polar type of substance. So, of course, I'm going to give you some examples. Examples are sugars. For example, glucose is a type of sugar. Salts, ions. Okay, remember we have various types of ions. All of these examples are examples of either large or polar substances. So please do not forget that that term polar means a charged substance, meaning it has positives or negatives or both of them attached to it. Oil does not like charged substances. Oil is not a charged substance, so it does not attract any sort of polar substances. And the last mechanism of transport that falls under the category of passive transport is osmosis, which we have already discussed in a previous page. So the first type major mechanism of movement across the membrane is called passive transport. Our second major type of transport across the membrane is going to be called active transport. Active transport is, in essence, the opposite of diffusion. That is the opposite of diffusion. What do I mean it's the opposite of diffusion? First, you have to understand what is diffusion. Diffusion is when molecules spread apart. We do not want to say molecules spread apart. We want to start integrating our scientific terminology. During diffusion, molecules move from an area of higher concentration to an area of lower concentration. Well, if active transport is the opposite of diffusion, that must mean molecules move from an area of lower concentration to an area of higher concentration. This would be like putting the perfume molecules back into the bottle after you sprayed them. This is when the substances move from an area of lower concentration to an area of higher concentration. This is said to be molecules are moving against the gradient. This is different than passive transport because during this type of movement mechanism, cells have to utilize their precious energy molecules in order to effect this type of movement. The best way to remember how active transport is different from passive transport is when a substance is actively transported, that substance moves against the gradient from low to high, and that precious energy molecule called ATP is needed. So I like to think of this as the three A's. Active transport against the gradient, ATP needed. Please do not forget that ATP is a very energetic molecule that is found in cells. And in order for substances to be actively transported, ATP needs to be broken down to provide the energy for that movement mechanism. Some examples of active transport are going to be, number one, protein pumps. For ion transport. The plasma membrane will have a protein embedded in the membrane, but it's going to be a special type of protein that will pump substances against the gradient. The first type of protein pump is going to be called the sodium-potassium protein pump. I would like you to review in your online textbook the sodium-potassium pump. But I will tell you that the sodium-potassium pump is an important protein pump because it is involved in at least one step of nerve impulses. Nerve impulses are basically messages from your brain that are directed along the brain cells, which are called the neurons. Of course, this basically controls every function that you have, including vital functions. So therefore, these sodium potassium protein pumps are absolutely critical to our survival. Okay, we need to be able to actively transport or force sodium and potassium ions to move against the gradient during at least one step that is involved in nerve impulses. The next type of pump is called a calcium pump. And again, you can review in your online textbook or you can Google. Okay, and I just want you to understand that calcium pumps are required during muscle contractions. There are many steps involved in a muscle contraction in terms of what's going on molecularly, but there's at least one step that requires the action of a calcium pump where calcium ions are moved against the gradient. The next type of active transport is a bulk transport. So think about bulk, that means large. And anytime you're moving something really large, either into or out of the cell, it is definitely going to require some energy of the cell. But one type of bulk transport is called exocytosis. Exo means out. X is out. This is going to involve vesicle transport like we discussed on the first page of our notes. Some examples of exocytosis are going to be mucus-producing cells, and neurons releasing neurotransmitters. Let's first talk about the mucus-producing cells. These cells, their whole goal is to make globules of mucus and release the mucus to the outside area in order to keep the linings of your body lubricated. So you're going to find these types of cells in any area of your body that needs lubrication. For example, digestive system, respiratory system. More systems, but we don't need to talk about all of it. I just want you to recognize what it is. So we have our mucus-producing cells. And another example of exocytosis is when a neuron releases neurotransmitters. All right, let's draw a picture of what it looks like. Here's your cell. And here's that vesicle that we talked about. That vesicle contains a lot of substances. For example, let's say that this is a neuron type of cell, and the vesicle contains neurotransmitters, or what's often called NTs. Neurotransmitters. NTs is the abbreviation. The vesicle will basically fuse with the plasma membrane, and that will ultimately create an opening for the neurotransmitters to be released out to the extracellular fluid. NTs released out. And the neurotransmitters will then go on to bind to the next cell to affect cell-to-cell communication. Okay, our next bulk type of transport is going to be called endocytosis. So on our last page, it was exocytosis. On this page, it is endocytosis. So instead of going out like what happens during exocytosis, there is an import of a bulk amount of substance. So endocytosis is when substances go in, and exocytosis is when substances move out. Three types of endocytosis to discuss briefly. The first one is called phagocytosis. So the best way to kind of describe phagocytosis is to make a drawing of it. But before I make a drawing, let me give you an example. Phagocytosis ultimately occurs when the cell kind of grows these two appendages like it's going to give a substance a very big hug. And when these appendages completely wrap around the substance and now the cell is hugging it, then it brings that substance inside. So I'm going to go ahead and draw a diagram to show what we're talking about here. So here's the cell, here's the ICF of the cell. And let's say there is a large substance out here in the ECF. So here's the ECF. And there's this large, bulky substance out here in the ECF extracellular area. So what happens is the membrane forms this little indentation to get ready to accept this very large substance. Here's our large substance. So the membrane forms a little indentation to begin to accept this large, bulky substance. And eventually, this substance moves all the way in. This indentation will eventually become the vesicle that will completely surround that large, bulky substance. And it will become a vesicle now containing that large, bulky substance. So here's that large, bulky substance, and the vesicle is formed. This is what happens during part of our immune system. And a great example of phagocytosis is the function of our white blood cells. We have a whole bunch of different types of white blood cells, but we do have some white blood cells called phagocytes. And basically what they do is they import germ cells, and maybe it is a bad bacteria or some sort of germ, and they bring it in order to destroy it. So in order for the white blood cell to destroy the germ, it has to first bring the germ inside of the white blood cell so that it can basically begin releasing specific chemicals out of that will break it down. This process called phagocytosis is often called cell eating. And if you think about it, that's kind of what the white blood cell is doing. The next type of endocytosis is called receptor mediated endocytosis. Receptor mediated endocytosis. This is going to involve a receptor protein, which is found on the surface of the cells. So again, let's go ahead and draw the cell. We'll draw the receptor. So here's the cell. Here's the ICF, the inside of the cell. And the first step involves a receptor. And basically, a substance will come along and bind to that receptor. It usually fits perfectly. So let's say this portion, okay, of that substance will move into the binding site and bind to that receptor protein. And that's basically going to tell this cell, okay, we want to bring this large substance into the cell. So then the cell, the membrane, will start to form a little indentation to accept this large bulky substance inside. And then a vesicle will be formed to accept this large substance. An example of this is unfortunately how some viruses infect our cells. Some viruses infect our cells by binding to receptors. And triggering endocytosis. It's not all viruses, but some viruses can do that in order to get inside of the cell and infect the cell. So that is just one mechanism that they may use. And it was all basically triggered when that virus basically attached to the binding site on the receptor protein. So it is all about the shape of it, and the shape has to fit into the binding site. Okay, our last example is called pinocytosis. And this one is the easiest, and it is actually just called cell drinking. So the cell is constantly surrounded by an ooey gooey nutritious fluid, and sometimes the cell wants to intake a droplet of that fluid and basically bring it into the cell, and that process is called pinocytosis. Please make sure to bring your handwritten notes to class for the diffusion and osmosis labs.